Hello, in this video we're going to be looking at general information for cellular respiration part 2, so let's not waste any of your time. Let's get right to it. Now when we're talking cellular respiration, what we want to think about is these opposite transactions, for lack of a better term, that are going on. We've talked about in the first one, if somebody's being reduced, somebody's being oxidized, okay, and that same with the exergonic, endergonic, storing potential energy, releasing potential energy. So we want to think about both here in chapter 7 and in chapter 8 that every time one of them, the building or breaking occur, you've got to be thinking that the other one also is occurring. Okay, if the glucose is being oxidized, then somebody's being reduced. Something is being reduced. Same with photosynthesis. When the glucose molecule is being built, it's being reduced. Say something is being oxidized, so you got to keep that in mind. So let's make sure that you do remember about the redox, reduction oxidation process. Remember that one, in this case we're going to be talking about glucose, the food molecule. So if one molecule is going to be losing the electron, as in glucose, the food molecule, then something is going to be gaining that electron. In this case, it's going to be the electron carriers that you'll be introduced to. We've seen them. We talked about them in 6, but now you're going to see what they're doing and why they're doing what they're doing. But remember, our oil rig, oxidation is the loss of an electron, oxidation, loss, oil, and reduction, gain, electron, rig, oil rig. So oxidation loses, reduction gains, and its energy value is what's being reduced. And the thing to think about it, if you want to follow the electrons, okay, see where the electrons are going, then follow the H's. Okay, if you want to follow the energy, then follow the H's. It's those H's that are going to be moved, that are going to be taken from the glucose molecule and carted away. And those electrons, those H's have the electrons, which is what this is all about. Ultimately, the electrons are going to be what activates the pumps on the membrane in chemoosmosis, but we'll talk about that once we get there. But follow the H's, follow the energy. Alright, so again, just reminding you about the redox situations. What you're going to be seeing is you're going to be seeing NAD plus and FAD, and we saw these two slides in chapter 6, but just wanted to remind you of what this stuff looks like. NADs, FADs are going to show up, and they're going to be carting away H's, headed off to the chemoosmosis electron transport chain, which we will get to. Now, and when they do this, okay, when they arrive at cellular respiration, they're going to be without, okay, but then they're going to take H's away, so they're going to be gaining electrons, which means they're going to be reduced. And when they're reduced, when they leave, they'll have those H's attached to their molecule. And the same thing happens to both. They gain the H's, so they're reduced. The glucose molecule will be oxidized at that point. Now, the one difference between the two, obviously besides the first letter, is that NADs can carry one H. Okay, FADs can carry two H's. So you can kind of think of these as, if you're going to move and you ask some friends for help because they've got trucks, they can load stuff up in the back, and one of your friends has a Ford Ranger, which can carry a lot of stuff, no doubt about that, but your other friend has an F-250, which can carry a lot more stuff. So they're both very helpful. They both can do what they do very efficiently. It's just the FADs can carry more. So just kind of keep that in mind. FADs can carry 2, and you'll always see that little 2 hanging off the H there, and NADs can carry 1. But so NAD, it's an oxidizing agent, and it just accepts electrons from the organic molecules. In this case, it's going to be the glucose, and when that happens, it gets reduced down to the NADH molecule. Okay, so here's what this looks like, and we want to tie this into all of our talk in Chapter 6, those enzymes and how important they are. So the blues, looking at the diagram beside me, starting from the left and going across, it's a little 3-picture action transition. So you've got the blue enzyme, okay, and this enzyme is the one that carries out the transaction. And when we look at the diagrams for cellular respiration, we'll see these enzymes. They'll be named, we'll point them out, but they always have those big, long, unpronounceable names, so they're easy to find in a diagram. But you've got this enzyme, and in the top part you've got the energy rich molecule, in this case glucose, and it sets into that one active site. And then NAD comes along and sets in the second active site. And of course the energy rich molecule glucose has those H's on there. And so the enzyme comes along, the redox reaction happens. And this is why they happen in tandem, because both molecules are on the same enzyme. And so the H's are removed. Now the thing I don't like about this diagram is it removes both H's but only attaches one to the NAD, so it should have been either an FAD or just do it twice, or leave the one H for next time. But you can see the final product, the H is now on the NAD there in the middle, and then as we know with enzymes, once it's done they release, but the product is a little different now than what it was. It has been converted, and again, then what will happen is another enzyme will come along, and that green energy rich molecule will now attach to that active site, and further conversions or changes will be made. But that's the idea of linking enzymes that we talked about in 6, showcasing what they're doing and how they're working this process. Okay, now this diagram is just a reminder that this is done in small steps, and with the idea of the enzymes, you can see another reason why it's done in small steps, because it isn't one major transaction that all that energy is released. No, each enzyme comes in and does its one particular job, and so it's a small steps. But again, trying to capture that energy. One way of thinking about it is, if you've ever eaten a Golden Corral, they're known for their buffets, obviously, and they'll have a meat area, and they'll bring out huge, like, it's a ham or turkey or roast beef, and they don't just take that roast beef and put it on one person's tray. No, they'll carve up a slice or a couple of slices and give some to one person, carve up a couple more slices and give it to somebody else, carve up couple more slices and give it to another person. So that process of carving off small pieces and putting them onto the trays or the plates for the people, that's a way of thinking about cellular respiration. You can think of the glucose molecule as that whole roast beef, And you can think about the carving off of a couple pieces as those enzyme actions, and you can think of the trays or the plates that the people have as the electron carriers, and then you can think of the people taking their plate or tray back to their table as the electron carriers going to the electron transport chain to drop off those H's. So that's a decent metaphorical look at the way things are happening here in cellular respiration. So hopefully that helps. All right, now one of the things that we want to think about is, for the most part, especially when we're involved, this all happens. It's aerobic respiration, means it happens with oxygen being involved. I mean, we breathe in oxygen. We've got oxygen readily available to help out with this process, and you're going to see that oxygen plays a major part in the grand finale of making ATP molecules. Matter of fact, oxygen has its own particular protein that it comes to work this process. But it also, this can be done, this process, in some organisms without oxygen. In other words, anaerobic respiration, to where the final electron acceptor is an inorganic molecule, not oxygen. In other words, inorganic, nitrogen, phosphorus, just some other element that isn't carbon related or have any carbon backbone to it. It's non-organic. It doesn't have those CHs, so it can be a nitrogen, as I mentioned, phosphorus, chlorine, whatever, just some other inorganic molecule. Now the third type, fermentation, and each of these will have their own slides later on in the later video. But fermentation can happen where, again, there's no oxygen involved, but the final electron acceptor is an organic molecule. In other words, it does have carbon involved, and the one we know this, we're familiar with fermentation. I mean, that's certainly how yogurt is made, cottage cheese, that kind of stuff, cheese in general, wine, that kind of stuff. It's all done through a fermentation process. But where we also know it is in our muscles, this can happen. So if you're working, whether you're working out or exercising or just working in the yard, and you do more than what your muscles are accustomed to, and they run out of oxygen, they'll continue to work, they'll continue to make ATP, but it'll be done without oxygen, and therefore fermentation will occur. And you'll know this the next day when your muscles are really sore. That's what has happened. Lactic acid has built up, causing that soreness, and you'll know that fermentation has occurred when you feel that muscle tension. All right, now the whole purpose of cellular respiration is to create ATP molecules, and we'll see that we've talked about ATP a couple of times. We looked at it in 6 pretty hard with the structure, but that adenine and those 3 phosphates and that ribose sugar, but the idea that cells are going to use ATP to drive endergonic reactions. In other words, the molecule is broken down, energy is released, that energy is used to make ATP molecules, and then the ATP molecules are used for endergonic reactions. In other words, the energy released helps build molecules. So again, it's that exergonic, endergonic, hand in hand. When one is happening, you know somewhere the other is happening. But ATPs can be made in a couple of ways, and we're going to see both of these. They can be made straight from energy generated from the breakdown of a molecule, the substrate. The substrate is broken down, energy is released, and that energy can be used to build the ATP molecule, and we'll see examples of that. The other way ATP can be made is oxidative phosphorylation, and that is just from the H's going through the ATP synthase protein, and we'll see that, and that happens during electron transport chain and chemoosmosis. So we'll see a bunch of substrate level phosphorylation, and then when we get to ETC, we'll see the oxidative phosphorylation. But the idea is just to make sure you understand the difference between the two. One, substrate level is done, the ATPs are made straight from the energy generated or released when glucose is broken down. The second, oxidative, is the ATPs are made as the H's go through the protein pumps. And here's how this looks. This is substrate level. And again, we just saw this kind of an example when we looked at the H's being removed from glucose. But here's what we've got. We've got your molecule that is being worked on. The PEP has a phosphate. The ADP at the bottom of the enzyme has 2 phosphates, so it's ADP, but it's being a third phosphate is being added, and again an enzyme is the one working that process. We know that we talked a lot about enzymes, bring down an activation energy and getting that job done, and when the transaction is complete, now you've got an ATP molecule with 3 phosphates. So you can think of the endergonic, exergonic reduction, oxidation process. The ADP molecule is reduced, the PP is oxidized, the PP is exergonic reaction, and the making of the ATP endergonic reaction, storing energy inside of that molecule. So again, I can't say it enough that those transactions happen in tandem, and that's something you need to be thinking about. If somebody's being oxidized, what's being reduced, and vice versa. If there's an exergonic reaction, where's the endergonic reaction occurring? Got to find it. Well, that is our video for part 2 of the general information for cellular respiration. When we come back next time, we're going to be starting the process of looking at glycolysis. So we'll see you then.